All right, guys, so here is the quiz key for chapters 9 and 11 quiz that you guys took last Friday. So I'll just go ahead and go through these questions. So if you did miss anything on the quiz and you're curious, feel free to check this video and you can see what you missed. So question number 1, we needed to organize the terms to indicate the correct order of the central dogma. So the central dogma starts with DNA. DNA is transcribed into RNA, and then RNA is translated into proteins. So that is the order of terms there for the central dogma of molecular biology. Question number 2, DNA replication is required to occur prior to transcription. This is false. We don't need to replicate every time we are transcribing. So that is false there. Question number 3. So there are 2 scientists credited with discovering the structure of DNA. Their discovery, though, was actually based on work by another scientist. So the scientists that are credited with discovering DNA is Watson and Crick. So that narrows us down to these 2 options here. And then their discovery is actually based on work by Rosalind Franklin. So that is the correct answer for this question. A lot of people said that Darwin and Alfred Russel Wallace were credited with discovering DNA. They are the ones who came up with the theory of evolution. So a different chapter for those 2 guys. Watson and Crick were our DNA scientists there, based on work by Rosalind Franklin. Question number 4, which of the following is not a component of nucleotides, which are the building blocks of DNA? So nucleotides have a sugar and a phosphate and a nitrogenous base. So they do have this inorganic phosphate, they have this nitrogenous base, but the sugar that they have is a deoxyribose sugar, not a deoxyglucose sugar. So this is what is not a component of a nucleotide there. Question number 5, so we've got a strand of DNA with this sequence that's replicated, and what is going to represent the newly synthesized DNA strand? So you can see that it starts with a 5' end here. So we are going to be starting with a 3' end going towards a 5' end. And then all of our A's are going to be pairing with T's. All of our T's are going to be pairing with A's. G's go with C's, and C's go with G's. So if we do all of that, that's going to get us this one right here. So these 2 are the same, but the reason that this one is right is because we have the 3' end over here and the 5' end over here. Question number 6, the nitrogenous base thymine is which type of base? So we have 2 types of bases. We have a purine and a pyrimidine. Monoamine is not a type of base, so purine or pyrimidine were the 2 real choices that you had here. And thymine is a pyrimidine, a single ringed structure. Purines have the double rings there. Pyrimidines have 1 ring, so thymine, that is one of our single ringed pyrimidine types of bases. Question number 7, which of the following accurately shows DNA base pairing and the number of hydrogen bonds? So there was a figure in the textbook and in the lecture slides that showed this. So the correct answer here is that adenine and thymine have 2 hydrogen bonds. Cytosine and guanine have 3 hydrogen bonds, and then adenine and thymine, just like we said above, those only have 2 hydrogen bonds. So these 2 answers are not correct. This is our correct answer here, that first choice. What molecule connects the sugars in a strand of DNA? So the sugars are connected in the backbone. So it's a sugar phosphate backbone. So phosphate is going to be what connects the sugars. Deoxyribose is the sugar. Ribose is the sugar in RNA. So those are not the answers. Phosphate is going to be the correct answer there. Question number 9. So we're talking about gene expression. What is that process? So let's see, we've got the process of transcribing DNA into RNA and then translating RNA into protein. That's our first choice. We've got transcribing RNA into DNA and translating the DNA into proteins, or transcribing DNA into RNA and translating RNA into chromatin. Okay, so we don't transcribe RNA into DNA. That's backwards. So we transcribe DNA into RNA. So we can already mark off question B as one of the ones that's right. So A and C both are transcribing DNA into RNA, but genes are expressed through proteins. So that first choice there, choice number A, translating RNA into proteins, that is the correct answer there. Those proteins are what are going to express those genes. Question 10, which enzyme is responsible for breaking apart the hydrogen bonds that hold the two strands of DNA together? So that is helicase. Helicase is the one that unzips the strands of DNA. So that is going to be the correct answer there. DNA polymerase is what's adding on the new nucleotides when we're building the new strands of DNA, but helicase is the one that actually unwinds it. Question number 11, we've got this picture here, and we've got one leading strand and one lagging strand here in our DNA replication. We wanted to say which one, the top strand or the bottom strand, is leading and lagging. So the top strand is our leading strand because that one is replicated continuously. And then the bottom strand here is our lagging strand because it's done in chunks, and then these chunks get connected together. So the top is the leading strand. Question number 12, what enzyme is responsible for adding nucleotides? So we talked about this when we were talking about helicase. This is going to be DNA polymerase, the one that's adding the DNA nucleotides to the chain, DNA polymerase. Question 13, which of the following occur in the nucleus? Select all that apply. So replication occurs in the nucleus. Transcription occurs in the nucleus. Translation does not occur in the nucleus. This occurs in the cytoplasm with those ribosomes. So translation does not occur in the nucleus. Question number 14, the flow of molecular information from genes to RNA to proteins is referred to as what? This is the central dogma. So that was kind of answered up in the first question, but that is the central dogma of molecular biology. Question 15, 3 types of RNA that are involved in gene expression. So all of these have mRNA. So that's the one that is transcribed from the DNA in transcription. Then we have tRNA in these two and xRNA in that one. xRNA is not a real thing. tRNA is the next one that we have. That is the RNA that brings the amino acids over to the ribosomes during translation. And then the last one that we have is rRNA. That is the RNA that is in the ribosomes. So B here is the correct answer. Question 16, list 3 differences between DNA and RNA. So DNA has deoxyribose, RNA has ribose, so difference in the sugar there. The deoxyribose missing an oxygen on one of those carbons in the sugar. The nitrogenous bases are also different. So DNA has thymine, RNA has uracil. And then the structure of them is a little different as well. DNA is double-stranded and RNA is single-stranded. So those are the 3 differences that I was looking for. You could have also talked about what DNA and RNA do and the fact that those are different. Their functions in the cell are different. But as long as you listed 3 differences, then you got full credit there. Question 17, what is the purpose of adding a 5' cap and a poly A tail to the single-stranded RNA molecule? So the purpose of this is to prevent degradation. Okay, if it doesn't have these end pieces on it, it will be degraded very quickly. We don't want it to be degraded until we're done with it. So we want to prevent that degradation by adding those things to the single-stranded RNA molecule. Question 18, so we have a DNA sequence here. We needed to write a corresponding RNA sequence. So C's always go with G's, T's always go with A's, and then G's always go with C's. But anytime we have an A, when we're talking about DNA, these A's and T's go together. Remember that RNA doesn't have the T's, so anytime we were supposed to have a T, we're actually going to put a U. So we're just writing this corresponding sequence there. Then we needed to use this genetic code to write out a sequence of amino acids that are coded by this piece of RNA. So this is our sequence of amino acids. The last one, this UAG, codes for a stop codon. So that is the last piece here telling us to stop, that we're done with this amino acid chain. Question 19, explain the difference between a point mutation and a frame shift mutation. So a point mutation happens at one point. It is a single base change which results in a change in one amino acid in the sequence. With a frame shift mutation, we are either having a base inserted or deleted, which changes every amino acid in the sequence after that one mutation. Okay, so difference there in the point mutation and frame shift mutation. Question 20, the development of evolutionary theory is credited to Charles Darwin and Alfred Russel Wallace. Those are the two people that are credited with that theory. Which of the following best describes microevolution? So microevolution is a change in allele frequencies of a population over time. That is the definition of microevolution. The change in individuals' traits as a result of natural selection. So microevolution is not just because of natural selection. There are other things that do microevolution. And then a change in an individual's traits. It's not going to change the individual. It works on the population level. Question 22, survival of the fittest, or the reproductive advantage in individuals who inherit traits that help it adapt to a changing environment, was something Darwin called natural selection. Okay, so we've got natural selection there as the survival of the fittest. Question 23, a heritable trait that confers on an individual a survival and reproduction advantage in the current environment is an adaptation. So that's an adaptation there, something that helps an individual survive and reproduce. Question 24, evidence believed to support the theory of evolution falls into which categories? So fossil record and homologous structures are in all of these. Those are both correct. We have biogeography in a couple of these. That's also another one that supports the theory of evolution. Astrobiology does not, but molecular biology does. So choice C is the correct answer there. Question 25, which of the following best describes the process that occurs when allele frequencies change in a population due to individuals migrating in or out of that population? So anytime there's migration, that is gene flow. Gene flow is the movement of alleles into or out of a population because of migration. Question 26, being a large population can help protect you from genetic drift. This is true, right? So genetic drift is a change in allele frequencies because of a drastic reduction in the population. If you have a really, really, really large population, genetic drift doesn't impact you as much as if you have a small population. Question 27, so we've got a population of Sasquatch in the Pacific Northwest that has variation in foot size. Variation is a normal distribution with a bell-shaped curve. However, females have begun to prefer mating with individuals that either have the smallest feet or the largest feet. Eventually, this will lead to a bimodal distribution instead of the bell-shaped curve. So this is an example of disruptive selection, where we start to have these two bell-shaped curves instead of just the one. If it was directional selection, all females would be preferring either the smallest, like all individuals prefer the small feet or all individuals prefer the large feet. But if they're preferring either the large or the small, that's disruptive. Stabilizing selection would be maybe females are preferring the moderate sized feet. So that bell-shaped curve is going to get more narrow. Question 28, southern sea otters off the coast of North America, west coast of North America, were hunted extensively for their fur. The original population was 150 to 300,000 individuals, but it got reduced to only about 1,000 individuals by the time hunting stopped. So as a result, the allele frequencies were probably different than the original population. So this is an example of genetic drift, right? We have this change in allele frequencies because of a drastic reduction in population. So when we talked about genetic drift, there were two different types. There was a founder effect or a bottleneck effect. This would be an example of a bottleneck effect. Question 29, two very similar rodent populations are found on two Pacific islands near one another. A scientist decides to mate some of the rodents and gets viable but sterile offspring. Why is this the case? So we've got a couple of options here. We've got sympatric speciation, we've got allopatric speciation, we have adaptive radiation. So anytime we're talking about speciation, if it's sympatric, there is no geographic isolation, but with allopatric speciation, there is geographic isolation. And the reason that these, when these rodents are mated together, their offspring are sterile is because they have finished the speciation process. They are two different species now as a result of allopatric speciation. Question 30. A scientist finds a new organism that may either be a bird or a bat, but it's not sure, the scientist isn't sure which group it belongs to. He thinks that the organism has analogous structures with bats, and homologous structures with birds, as suggested by genomic data. The organism has one trait that is shared with bats, but not with birds. So which group should he choose to put the organism in? Which group should he classify it with? So homologous structures are structures that have one evolutionary origin, but they may have kind of diversified, but have the same basic structure. And then analogous structures have separate evolutionary events that have converged on similar structures. So this is the result of convergent evolution. So if we're thinking about how to classify this, we want to classify it with the homologous structures, talking about the same evolutionary event. So we're going to want to classify it with birds because of the homologous structures. That narrows us down to A and B. The trait only shared with bats is derived, or the trait only with bats is ancestral. So the trait only shared with bats is derived. That's something that happened. Maybe there was some sort of evolutionary pressure that caused this type of organism to develop a trait that bats have also. Maybe bats are under the same evolutionary pressure. It's not ancestral, because that would mean that there was a common ancestor of bats and this organism that have it. This is a derived trait, so we're going to choose A there. Question 31, the wing of a bird and the wing of an insect are an example of analogous, homologous, or isologous structures. So the answer there is analogous, right? This is not one common ancestor had wings and then birds and insects split from that ancestor. That's not what happened. There were two separate evolutionary events that developed wings, one for birds, one for insects, and they converged on these similar structures. These are analogous structures. Question 32, two organisms mate and have offspring. How could you test to see if the parents were the same species? So we are going to test to see if the offspring are fertile, right? If the offspring are fertile, then they are not two separate species, okay? Right, that's one of the things that separates one species from another species. If they can cross and have fertile offspring, maybe speciation just isn't quite complete yet. Question 33, so we've got a population of 10 squirrels. In this population, having a bushy tail is dominant to having a slick tail. There are 5 homozygous dominant squirrels, 3 heterozygotes, and 2 homozygous recessive squirrels. We want the allele frequencies for the big T, the bushy tail, and the little t, the slick tail. So if we count up the number of big T alleles, there are 20 alleles total in this population because each individual has 2 alleles. So 10 squirrels, 2 alleles each gives us 20 total alleles. The big T's, we can count these up. So in 5 homozygous dominant squirrels, there will be 10 big T's, right? They each have 2. In the heterozygotes, they each have 1. So that's 10, 11, 12, 13 big T alleles. So if we do 13 out of 20, that's 0.65 is our allele frequency for the big T. And then our little t, if we look at our homozygous recessive squirrels, there are 2 of those, so each of those have 2 alleles. That would be 4 alleles. And then 3 little t's from our heterozygotes gives us 7 out of 20, or 0.35. So one summer it's really hot and it becomes advantageous to have a slick tail. It's too hot for a bushy tail. At the end of the summer, we've got a different set of squirrels than we did before. 2 homozygous dominant, 2 heterozygous, and 6 homozygous recessive. So we needed to calculate these new allele frequencies. So that's down right here. So if we count up our big T's, we've got 6 total. We count up our little t's, we've got 14. So the frequencies are 0.3 for the big T, 0.7 for the little t. Question 34, so list 3 of the 5 mechanisms for evolution. You can choose one and explain it in as much detail as you can. So the 5 choices you had were mutation, gene flow, genetic drift, natural selection, and sexual selection. Those are the 5 choices that you have. So you could have chosen any of these to explain. So I kind of just gave you credit as appropriate if you explained it correctly or not. So feel free to let me know if you have questions about these if you missed credit on any of those. Question 35, explain the difference between homologous and analogous structures. We kind of did this up in a previous question, but basically those homologous structures have one evolutionary origin and the same basic structure, but they've diversified into different forms. The classic example of this is the mammalian forearm, because we've got forearms like we've got forearms like cats, forearms like horses and bats and whales, lots of different forms, but when you look at the skeletal structure of those, it's the same basic structure. That's a homologous structure. Analogous structures look very similar, but have separate evolutionary events that gave rise to these structures, the result of convergent evolution. So wings of birds, wings of bats, wings of insects, those all evolved at separate times, but it was advantageous to be able to fly. There was the same evolutionary pressure on these species. So there was this separate evolutionary event for each of those groups that gave rise to similar structures. And then question 36, so Joe Exotic in Netflix's Tiger King had ligers, a lion-tiger hybrid in his Oklahoma zoo, quote unquote. Why don't we find ligers in the wild? So this is because of a geographic reproductive isolating mechanism. This is one of the pre-zygotic reproductive isolating barriers, right? Lions and tigers don't live in the same place, so they don't encounter each other in the wild, so that's why we don't find ligers in the wild. Okay, so feel free to take a look through your answers, see what you missed. Let me know if you have any questions. And then I will post the next key for the next quiz as soon as you guys have taken that one.